Hello, and welcome to Lab Study Guide number 3. This is all about tissues. Now, this is a very critical chapter in the textbook and a very critical piece of lab area for us. So there's going to be a lot of overlap between the lab and the lecture here. And the reason for that is because this chapter and this particular subject is so important to us. So understanding tissues, the different types of tissues, their form, their function, and their structure, all of that is extremely important. And in fact, I could say that it's the foundation of everything that's going to come after this. So when you look at all the different organ systems, you're going to have to understand what tissues are, what are the different types of tissues, how are they structured, and how they function. So we're entering into a very critical time, and actually, this is probably the first area that we've studied so far where most of you are on unfamiliar ground. Up until now, we've been basically reviewing information that you should have gotten in your general biology course. So this marks a beginning of new information, something that you haven't yet, most of you anyway, haven't yet been exposed to. So let's talk a little bit about how this is going to work. So instead of having one or two long lectures, I'm going to break this up into shorter lectures. And because I want to break it up into distinct parts so that when you go in and look at these things, you can identify specific areas where you're perhaps a little hazy or not quite strong on, and you can go back and just cover that particular area. So let's begin by looking at the lab study guide and what it tells us. So the information that's covered is tissue types, form, function, and structure. Core concepts are tissue, structure, and function. And then I've given you a series of things that you should know. So name the 4 primary tissue types and their general functions. Compare and contrast the 4 primary tissue types. Describe and understand the basic structure and parts of epithelial, connective, muscle, tissue, and nervous tissue. Identify examples of epithelial, connective, muscle tissue from slides and images. So we are not obviously going to be looking at slides, but you will have a lot of images available to you. And hopefully through practice, what you'll be able to do is look at an image and say, that is epithelial tissue, that's simple squamous epithelial tissue, or that's adipose tissue. So you'll be able to just recognize by sight that type of tissue, and then also connect to that tissue the important facts that are associated, how it functions, where it's found in the body, and so forth. Describe examples of the location and function of epithelial, connective, and muscle tissue covered in the lab. And then I've referred you to some pages here in the textbook. Okay. So what we're going to do today is we're going to cover the 4 basic types. So we're going to look at the 4 basic types of tissues that we find in the human body. And then I'm going to focus on epithelial tissue. So this chapter or this lecture for the lab will introduce the subject of tissues, and then we'll spend some time looking at epithelial tissues. The following lectures will deal with connective tissue, as well as muscle tissue and nervous tissue, and probably some additional information in terms of looking at images and being able to analyze the image and understand what you're looking at. Okay, so let's begin that discussion then. And let me just remove the study guide, all of the pages, so we can start with then our notes. So this is tissues. Okay. And this is going to be lab study guide number 3, I believe it is. Number 3, that is correct. Okay, all right. So there are 4 primary tissue types. These include, number one, epithelial tissue. Number two, connective tissue. Number three, muscle tissue. And number four is going to be nervous tissue. These are the tissues that your body are composed of. These four basic types of tissues. And in terms of comparing and contrasting them and looking at their structure and so forth, what I'm planning to do is take a single lecture presentation, and especially with the first two, epithelial tissue and connective tissue, so devote entire sort of lecture to each of those. I will probably combine these two into a third lecture, and then hopefully I'll also have time for a fourth or fifth lecture in which I present images to you and I walk you through those images so you can begin to form what I call search images. So the way your brain works is if you're looking for something, then you have an idea of what that thing looks like in your mind. You have an image of that thing in your mind. Say you're looking for your car keys. You have an idea of what they look like. And as you search the environment with your eyes, you're trying to match objects that are in that environment to that perceived picture in your brain. And when those two things match, your brain says, I have found my keys. So if you don't have a search image, you'll never see what it is that you're looking for. So one of the things that I want to do in these lab presentations is give you that search image for epithelial tissue, connective tissue, muscle tissue, as well as nervous tissue. So that's what we're going to do. We're going to be talking about the functions and structure and other things as we go through. And I will develop all those ideas in much more detail in the lecture. So as I said in the beginning, a lot of overlap here. And that's good because this is new information. This is new territory for us. And if we get things repeated over and over again, it can only be to our advantage. All right, so let's begin with epithelial tissue. So epithelial tissue epithelial tissue. So what we can say about epithelial tissue is it's broken down into two major sort of different types of epithelial tissue, two different categories of epithelial tissue. So the first category is going to be covering and lining epithelial tissue. Covering and lining, what does that mean? It means that it's going to cover body surfaces hollow tubes and organs, or I should tell you body cavities. Let's put it that way. Okay, so it covers body surfaces, hollow tubes, and body cavities. So the skin is a perfect example of epithelial tissue covering a body surface. The lining of blood vessels, hollow tubes lined with epithelial tissue. Body cavities, we've already talked about this to some degree. When we looked at body cavities, we looked at the parietal layer of the peritoneum, for example, but we can also apply that to the stomach. The stomach is lined with epithelial tissue. That's a body cavity. So any body cavity, any body surface, more than likely, and I can't think of a surface that wouldn't be, is going to be covered with epithelial tissue. So covering and lining epithelium is one type. A second type is glandular. epithelium. So glandular epithelium is just what it sounds like. This produces our glands. So here we're going to be looking at glands, and all kinds of glands. The thyroid gland, the adrenal gland, the liver, which is a gland. You name the glands, sweat glands, the glands that produce earwax. All of that is epithelial tissue that has organized itself into some sort of gland. So glands of all kinds, sweat, body oils, a variety of different ways in which this is manifest in your body. So glandular epithelium. In our presentation today, this is where we're going to be focusing, this area right here, the covering and lining epithelium. We're going to save that discussion, glandular epithelium, for the lecture. So I'm focusing here in our presentation in the lab, and I'm going to include this as well as that when we get into the lecture presentation. So let's talk a little bit about the functions then of epithelial tissue. What does it do for us? How does it contribute to homeostasis? So let's talk about some of the functions. In fact, all of the functions. Very briefly, not in a great deal of detail, but just so that we know. Number one, protection. So epithelial tissue can provide and does provide protection, and the classic example is your skin. Okay, so that's a real clear example of how epithelial tissue is protecting you. In fact, as you will learn when you get to the chapter on the immune system, this is the best and major protection you have against disease, is your skin. So protection is a huge part of the function of epithelial tissue. Not confined just to the skin, by the way. Every surface that's open to the outside world is protected by epithelial tissue. And we'll get into the details of that a bit later on. So secretion is another function. Secretion, such as hormones or enzymes. A number of different things are secreted by epithelial tissue. Hormones are clear. Secretion, enzymes are being produced in, for example, the digestive tract and secreted into the small intestine and into the stomach to digest your food. So secretion is a big function for epithelial tissue. Number three is excretion. So let's get this down. Then I want to explain the difference between excretion and secretion. So excretion is a function of epithelial tissue. One good example is carbon dioxide. That's an excretion that your body produces. And the cells that are doing that, of course, are in your lungs, and those are epithelial cells. So what's the difference between secretion and excretion? Secretion is a substance that your body is going to use. It's a useful substance. It's something that has a useful function in your body, such as enzymes, such as hormones. Okay? Excretion is waste. So basically what we're talking about is we're getting rid of something that you don't need. It is a waste product, and you excrete that out to the outside world. Carbon dioxide is a waste product. It's produced as you burn your food, and the carbon that's in your food is right there. So when you eat your candy bars and what have you, the carbon that's in those candy bars are leave your body as a gas, carbon dioxide. So you literally transform solid food into a gas and get rid of the waste products through carbon dioxide. So kind of interesting. All right, well, let's go on with some other functions. There are other functions. So we talked about excretion. We can talk also about, number 4, absorption. So absorption plays a big role in epithelial tissue. Nutrients is a good example. Ions, a number of things can be absorbed across the epithelial layer. Of course, this is obviously in the digestive tract. But there are other places, in the kidney, for example, where absorption is taking place across some epithelial surface and through epithelial cells. So absorption is a big function. Number 5, filtration. Filtration is taking place primarily in the kidneys. That is to say, where we see epithelial tissue performing this function of filtration is in the kidneys. And this is happening when you filter your blood. So you will learn about this in 40C when you get into the urinary system, which is a fascinating system. It's one of my favorite systems, and simply because of what's happening in the kidneys. The physiology of the kidneys it's pretty amazing. I really loved to teach that subject when I was teaching 40C because it was just such an amazing place to see all of the physiology. Anyway, long story short, kidneys are involved in filtration, and the filtration surface, the surface over which you filter your blood, is epithelial tissue. Okay, so filtration is a function. And the last function we will list, and in fact the last function that we need to list, is sensations. So we can turn to our skin again. So this is, of course, part of the function of the skin, is that sense of touch that's coming out of epithelial tissue. Also, when we combine nervous tissue with epithelial tissue, we get our special senses, such as the sight, hearing, smell, taste, all those special senses that you can name. Those come out of a combination of epithelial tissue and nervous tissue. All right, so those are the functions. Let's give some consideration to the characteristics. So when we look at epithelial tissue, what kinds of structures, what kinds of organization do we see? And that's going to be the characteristics of epithelial tissue. So let's turn our attention to that. So the characteristics of epithelial tissue. Right. Number 1, when we look at epithelial tissue, what we're going to see is a lot of closely packed cells held tightly together. There's going to be very little extra cellular stuff. And I'm going to put little parenthesis marks there because that's a scientific term, stuff. As soon as you do that, it makes it scientific. All right, little extracellular, outside of cells. So mostly what you see when you view epithelial tissue, for example, under the microscope, is going to be a lot of cells that are closely packed together. They're held tightly together because cell-to-cell connections are done through tight junctions. So we're going to see tight junctions between cells. This is a way we're going to weld one cell to another. We'll be talking about these, and it's the first thing we start off with in the lecture on tissues, is we're looking at all the different types of junctions and connections between cells, and tight junctions is going to be one of those, and we'll take a close look at that there. So tight junctions. Number 3, we're going to have a supporting basement membrane. A supporting basement membrane. And so very quickly, we're going to come back and develop this idea both here and in the lecture. If you have a cell, a series of cells, as you'll see, they're held together, tightly packed together. Supporting underneath is going to be a basement membrane. So this structure underneath is the basement membrane. And as I say, we're going to get into the details there for sure in the lecture and also in this presentation a little bit later on. So this idea of a basement membrane, we'll look at that. So supported by basement membrane. Number 4, avascular. What does that mean, avascular? It means there are no blood vessels. In epithelial tissue. The blood vessels are close by, no doubt. They have to be in order to keep the cells alive. And so what you typically see is the blood vessels here just underneath the basement membrane, but not in the cells, not up here in the cells, but associated with the basement membrane. And so within the epithelial tissue itself, no blood vessels. So it's considered to be avascular. All right. Number 5, as I mentioned before, when we associate this with nervous tissue, we get the special senses. Eyesight, hearing, taste, smell, all of these things are a combination of nervous tissue and epithelial tissue. Okay. Number 6 usually has a high rate of mitosis. Cell division is very common and happens often in epithelial tissue. And there is good reason for that. We'll talk about that more later on. So a lot of mitosis, a high cellular reproductive rate in epithelial tissue. Number 7, and I'm going to use the next page for number 7, so I'm going to skip to the next page now that we have this. Number 7 on the hit parade is the cells have orientation. And let me explain what that means by way of a very simple diagram. I'm going to draw one single cell, one single epithelial cell, and there it is in all its glory. And remember, we have this basement membrane, always the basement membrane. Okay? And now we can identify, because this sits on a basement membrane, it has orientation. And that is to say that this surface that is sitting on the basement membrane is referred to as the basal surface. This surface and that surface referred to as the lateral surface. And there at the top, we find the apical surface, or apical surface. Okay. So cells have that kind of orientation. And that comes out of, please notice, that comes out of the fact of that we've got a cell connected to a basement membrane. So that gives that cell orientation, as opposed to a cell that's disconnected from anything and is just kind of all jumbled up together with other cells. Being able to identify where the basement membrane is when you're looking at images will be important, and I will talk about how that's important later on when we look at how we name different types of epithelial tissue. Okay, so that's number 7. Number 8 is it's going to be found arranged in sheets of cells. Now this is for lining and covering epithelium. Okay, so the arrangement of the cells is always in sheets, not with glandular tissue, not with glandular epithelium, but with the lining and covering epithelium, they're going to be arranged in sheets of cells, and that will become very apparent as we look at different types. So these can be a single layer, or it can be multiple layers. We'll get into that detail in just a bit. Okay. So, with that in mind, what we're going to do now is we're going to look at how we name epithelial tissue. So how is it organized? Because there's not just one single type of epithelial tissue. There are several different types of epithelial tissue. How do we organize that? How do we organize all the various types of epithelial tissue? And that's what we're going to look at next. And then we'll start looking at some examples of epithelial tissue. So this is going to be labeled organization of epithelial tissue types. Okay. We're going to do this by using two characteristics. So two characteristics are going to be used to identify the various different types of, and this is, again, I must emphasize, this is lining and covering epithelium. This is not a discussion of glandular epithelium at this point. So number one is going to be cell shape. What are the shape of the cells? And they happen to fall out into a number of different very discrete and easily recognized shapes. So cell shape, okay, that's one characteristic. The other characteristic that we're going to use is the number of layers of cells. Okay? The number of layers of cells. And I'll describe what that means in just a bit. So let's start with the cell shape. Cell shape is going to be one of these types or shapes. Number one, they can be flat, in which case we're going to use the term squamous. To describe that. Flat, squamous, okay? Don't use flat, that's its shape. We're going to describe that shape as squamous. That's the name. So number two is going to be cube shaped. And here we're going to call that cuboidal, very conveniently. Okay. I'll show you these shapes in just a bit. We're just getting it down in our notes right now. So cell shape, flat, squamous, cube shaped, cuboidal. A third shape is going to be column-like. And here this is going to be called columnar. So those are the three basic shapes. Now I'm going to put a little line, a little squiggly line here, because I'm going to introduce another cell, not so much shape, but there's no real good place to put this category. So I'm going to put it here. It does make sense to fit it here. It certainly doesn't make sense to put it under number of layers. But this is, let me write it down and describe it a little bit, and then you'll see what I'm talking about. So this is called transitional. These are transitional cells. And what these things can do, what these cells can do, is they can change shape. So they don't assume one particular shape. They shape shift depending on the environment. And typically what you're going to see is a cell that can go from cuboidal shape to squamous and then back again. So these cells can transition between these two basic shapes. And we are going to see this type of cell in organs that are going to expand and contract on a regular basis. And the best example, in fact, the one of the greatest examples, and where we find most of the transitional epithelium in our body, is the urinary bladder. If you can picture what that structure does over a 24-hour period, it fills up with urine, you void, it's empty, it fills up again, you void, it's empty. So the urinary bladder is like a balloon that expands when it's filled with urine and then collapses when you void that urine. And so the walls are constantly being stretched, and we have to have some way to increase the surface area of the lining of this organ as it expands, and then it has to be able to squeeze back down into a smaller surface area, back and forth. And transitional cells are going to give us that characteristic, that function. So cell shape, flat, squamous, cube-shaped, cuboidal, column-like, columnar. And then there is transitional. All right, so that's cell shape. What else have you got for me? Well, let's talk about layers of cells. Layers of cells. So here we're going to have a couple of different possibilities. We can have a single layer of cells, in which case we're going to call that simple. Or we can have multiple layers. So it can be multi-layered, in which case it's called stratified. Okay. That's pretty simple. A single layer of cells or a epithelial tissue with many layers of cells. So either simple or stratified. Unfortunately, they've thrown in a little curveball on you. And I'm going to put another little squiggly line here. There is another possibility, and that's going to be called pseudo-stratified. Epithelium. Pseudo, meaning looks like it's stratified, but in fact it is not. So looks multi-layered, but is not actually. Okay. How is that possible? Well, a very little quick drawing can help us understand how that's possible. So most of the cells are going to be more or less cuboidal, and then in between them there will be cells that are shorter, and you're going to see something like this. And I'm not going to go ahead, I'll put in another one, I guess. The little dots I'm showing you, of course, are the nuclei. So one of the things that you're going to be able to do to tell whether you have a single layer or simple or stratified is count the number of nuclei. So if you can identify, and this is why knowing where the basement membrane is so important, if you can identify multiple layers of nuclei, that indicates you're looking at a multi-layered or stratified layer of epithelial tissue, because each one of those would represent a cell, okay? And so you would have them stacked on top of each other. multi-layered, and you would see this effect, nuclei stacked on top of each other, because the nuclei are very prominent. Okay? If, on the other hand, you had just a single layer, you would only have a single layer of nuclei. Okay, so that's simple, that's stratified, but look at this. If you're looking at this, you'd say, oh, look at that. We've got some stacked nuclei. That must mean that it is stratified, but it is not. It only looks like it's stratified because some of the cells don't reach the apical surface. Do not reach the apical surface. They are shorter than the others. Okay, and that is pseudostratified. All right. This is a difficult one to spot through the microscope. You really have to develop a good eye for that and get lots of practice. We'll talk about that later. All right? Alrighty. So let's reinforce some of the ideas we've talked about using some nice drawings here. So here is our first image, and this is going back to what we had talked about before. So if you can see, we have closely packed cells, and they are attached to a basement membrane. And because they're attached, we can identify specific orientation. The basal surface, the apical surface, the lateral surfaces. We also see that the blood vessels are not in the epithelial layer. There's not a lot of extracellular material. And in this case, they're showing us nerves that are penetrating into the epithelial layer. So this might be an example of, say, the olfactory membrane in your nasal cavity, being able to breathe. So materials that are in the air would be taken in by these cells, or actually they wouldn't be taken in, but they would bind to receptors on the cell surface, and then there would be a series of chain reactions that cause this nervous tissue to fire off action potential into the brain, and you have a sense of smelling a rose or whatever. Okay, so that's a picture of typical layout of epithelial tissue. Let's talk about these cell shapes, and we'll start off with the cell shapes. So I'm going to hide part of this. So we're going to just focus on that. So cell shape. Okay, so we get these flat cells. Okay. We get the cuboidal shaped cells. We get the column-like cells. So squamous, cuboidal, columnar. So those are the three different shapes. So they're not showing us transitional in this drawing, and that's probably good. We will look at transitional epithelium a little bit later on, but just so as not to confuse you, we have these three basic shapes. The flat, the cube, and the column. Squamous, cuboidal, columnar. Then we have different shapes, different layers. So here we have a single layer that's called simple. Here we have multiple layers that's called stratified. And here you can see I got 1, 2, 3, 4 layers. But before I could count those, I had to know where the basement membrane is. Because observe, if I do this, and remember when you're looking through a slide, it's not going to be nice and neat. It's going to be at different orientations. I might be able to say, oh, look at this. 1, 2, 3, 4, 5, 6. Oh, that's stratified. No. Why? Because I have to find that basement membrane first and then count up from that point. And if I do that, I only have a single layer. Okay. And this, I know at this point, maybe some of you are getting kind of concerned. Can I find the basement membrane? Yes, you will find the basement membrane. That's what all the search images are going to be all about, is helping you to do that. And after we get through this, you'll be experts at identifying the different types of epithelial tissue as well as connective tissue. Okay? So how do we combine this together? Well, let's take these examples and name some tissues. So I'm going to say that I have a single layer of cube-shaped cells. So that would be called simple cuboidal epithelium. Simple cuboidal epithelium. One layer, simple, cell-shaped cuboidal. What about this? Well, I have multiple layers. So this would be stratified cuboidal epithelium. Multiple layers, cell shape, stratified cuboidal epithelium. What would you call this? If you said simple squamous epithelium, you would be correct. That's a single layer, simple, cell-shaped squamous, simple squamous epithelium. What would you call this? If you said simple columnar epithelium, you would be absolutely correct. You have a single layer, simple, and the shape columnar, simple columnar epithelium. Okay? So hopefully you get the hang of this. And of course I can start to mix and match. I could literally have stratified columnar. Clearly you can see stratified cuboidal, certainly stratified squamous. But if I said stratified squamous, your expectation is you're going to see a lot of flat cells stacked on top of each other. And that's exactly what you would see. Okay. All right. So with that in mind, we're going to run through the different tissue types. So now what I've done in this lab is I picked out not all of the tissue, not of all the epithelial tissue that there is out there in the human body, but what I've identified is the most common type of epithelial tissue you're going to run into. Yes, there are other kinds. I will cover those very briefly in a lecture. But in the lab here, I'm focusing on that type of epithelial tissue that you're most likely to run into and to be interacting with in terms of physiology and other things of that nature. So first is this. And as you can see, what we're talking about here is simple squamous epithelium. Simple squamous epithelium. So a number of things. First of all, I want you to bring your attention over to this drawing. They're identifying something called the peritoneum. We know what the peritoneum is. Remember, that is that serous membrane that lines the abdominal pelvic cavity. It sits on the wall. It covers the wall like wallpaper, and it covers the organs. Whether it covers the wall or organs doesn't matter. It's the same type of tissue. It's simple squamous epithelium backed up by a very thin membrane, basement membrane, I should say. Okay, so simple squamous epithelium, and this is just an example of where we would find that. It is all over the body. They're just using that as one example. Now, here is a problem that students will identify, or they'll begin to be a little concerned about, because I've got a single layer of cells, and if I look at it from the side here, it's clear. But most times, when you look at this tissue, you're actually looking down at it as if you're flying over it with like a helicopter and looking down on the earth. And now you're saying to yourself, well, how do I know there's not other cells underneath? Because I'm only looking at the surface. I'm not looking at the side. I can't count those nuclei because I cannot really see the basement membrane. And that's exactly this view. This view is a helicopter flying over this and looking straight down and snapping a picture of this layer of cells from the top. And you can see a couple things. First of all, you notice that the arrangement, the way they're interconnected, is irregular. That's by design, because if they were all lined up like you would line up tiles on a kitchen floor, that would create a line of weakness. And so if a tear developed, it would just follow that line right straight through the epithelial tissue. But in this case, you can see this fact that it has all of this angle to it. It's harder. So if a tear starts here, it would tear, but then it would stop right there, hopefully. And so only a short area. It wouldn't travel through the full length. So this irregular pattern that you see, that's deliberate. Also, remember that this is a very thin layer. So if there were other cells underneath it, you would see ghost-like nuclei underneath. But what you're seeing is no ghost-like nuclei. You're only seeing that nucleus for this cell, that nucleus for that cell. You're not seeing nuclei that appear to be stacked on top of each other, like a stack of coins that are out of cattywampus. No, that's not what you're seeing. You're only seeing that cell associated with, that nucleus associated with that cell. If there were another cell, if there were another flat cell underneath it, you would see the shadow or the ghost of the nuclei of that one and probably several layers as well. So you're only seeing a single layer here. And that's the best I can tell you as far as simple squamous epithelium. Okay? The next type of tissue is simple cuboidal epithelium. Simple cuboidal epithelium, and that means we have a single layer of cube-shaped cells. And I draw your attention to this area of the picture. So here we are in the pancreas. As you will learn when you do digestive system in 40C, the pancreas has some ducts that are collecting all of the digestive enzymes. So the pancreas is producing a number of digestive enzymes which are going to be dumped into the small intestine. And they're collected in a series of ducts. And those ducts, as you can see here, are lined with epithelial tissue. And you can see the squareness of these cells. Now, here's something else that's going to be important for you to see. Or to utilize when you're looking at this tissue. You will notice that I cannot see a line. I don't see a square here. I don't see any real squares like they're doing in the drawing. I don't see this kind of square design. But what I see are very oval or round nuclei. And if you look carefully here in the drawing, so cuboidal shaped cells typically have a very round or oval shaped nucleus that takes up almost the entire inside of the cell. And so if you see that pattern as you're seeing it here, you can pretty well bet that what you're talking about is cuboidal shaped cells. Okay, so cuboidal shaped cells based on the shape of the nucleus and the size of the nucleus relative to the rest of the cell. You see that nucleus is taken because the basement membrane is right here. And there's the apical surface. And so this cell is a very short cell. And the nucleus takes up almost the entire space of that cell. And it's round and oval. So that's a key thing that you should be looking for as you look at the images of epithelial tissue. Okay. All right, so this is, and we'll put this in English so we can read it. This is simple cuboidal epithelium. Simple cuboidal epithelium. And they put this little thing in front of it, non-ciliated. And that says exactly what it sounds like. No cilia. And you're looking at this and say, well, I don't know. Maybe there's cilia there. Look at this. Maybe there's cilia there. No, that's called microvilli. And these are little finger-like extensions of the cell. So if I took one of these cells and I blew it up so you could see it in a drawing, what you would see is this. Look like little fingers sticking out the top of that cell. And here's my basement membrane. Okay. These are the microvilli, little finger-like extensions of the membrane. And remember when we talked about, in the last lecture, we talked about the cytoskeleton and the microfilaments, intermediate filaments that were making up the skeleton. These shapes are maintained by those filaments that we were looking at. Okay, so we'll come back to that. So these are just little finger-like extensions. And when you look at this tissue underneath the microscope, it's got this fuzzy edge to it. It's not a crisp edge. Got this very fuzzy edge to it. And that's because the microvilli make that a fuzzy edge. Okay, so they are long column-like cells. Okay, And interspersed is a special type of cell called a goblet cell, which is producing mucus. And here you can see that here. So these goblet cells always show up as like balloon, usually a light colored balloon structure amongst the cells. And you can see that clearly. That's a mucus producing cell. So this is part of your digestive tract. It's the small intestine. And the small intestine are lined by simple columnar epithelium with the microvilli. Why do they have microvilli? To increase surface area. So the small intestine does about 80% of the absorption of your meal. It's a lot of work that has to get done in a relatively short tube. And so to increase that efficiency, you're going to get a couple of mechanisms that increase surface area inside the small intestine. You're also going to be protecting the surface with mucus. You're mixing the mucus with your food, and so that's why we got the goblet cells. Now here I want to call your attention to the shape of the nuclei. See, the nuclei are more elongated. They're not that round nuclei that we saw with the cuboidal shaped cells. They're elongated. And typically you'll find them closer to this membrane, the cell membrane, okay? Excuse me, the basement membrane. So they tend to cluster at the basement membrane, as you can see right here. So there's a lot of space above them. And they're oval shaped, and so the basement membrane would be sitting right about here, okay? Right about there. And so that's one way you're going to be able to distinguish this from cuboidal shaped cells, is the shape of the nuclei, their position down at the bottom of the cell as opposed to in the center of the cell. And also this is a good opportunity to point out that you can see little ghosts here, right here. There's a nucleus, and then there's a nuclei behind it. So I know there's another cell behind this one. Just as you can see in the drawing, there's this one, and then there's a cell behind it. This one and a cell behind it. And these cells are so thin that when you're looking through the microscope, you're actually going to be able to look through one cell and see the ghost outline of the one behind it. And so you're going to get, here's another example, you get this ghosting effect. So when you think about that simple squamous epithelium, and you're wondering, well, how do I know that that is a single layer in English? There's no ghosting here. You don't see nuclei behind other nuclei. Okay? So that's going to give you a good, strong indication that you're looking at a single layer and not multiple layers. Okay. That's simple. Simple columnar epithelium, non-ciliated. Then we have stratified squamous epithelium. So a lot of flat cells stacked on top of each other. And by the way, when we talk about stratification, we're not talking about just 2 layers or 3 layers. Generally, it's a lot more than that, particularly with stratified squamous epithelium. Many, many layers. So again, this is the vagina. And so when we see stratified squamous epithelium in any part of the body, what we're concerned with is protection. Protection. The skin is going to be stratified squamous epithelium. Here in the vagina and a number of other areas, we're going to see stratified squamous epithelium. It's all about protection. We're not going to be worried about absorption or any filtration or anything else. It's all about protection. And so we're going to line those layers where we need a lot of protection with the stratified squamous epithelium. So if you can look carefully here, there's a couple things that I want to point out, and you can see it more closely right here. You can see it more accurately right here. Take a good close look at the cell shape as it moves up to the top surface. These definitely do not look like they're flat. These don't look like they're flat. It's not till you get to around here that they begin to take on that flat shape. And it's not actually till you get to the very top layer that they're really, really flat. So why are we calling this stratified squamous epithelium when in fact you can see that not all the cells take on that shape? They're not all flat. And the answer to that question is this outer surface is the functional layer. This is where we're getting protection. This area right here is where we're getting protection. What's happening down here is cell division, maturation of the cells. We're going through a developmental period right here. And as that happens, the cells are going to get flatter and flatter. They're going to die. And then we have multiple layers of dead cells at the top layer here. And that's where we're getting our protection. So the functionality of this layer of cells is up here at the top. And they are flat, so we call it stratified squamous epithelium. Okay. Finally, the last type of tissue, there we go. That is the last type of epithelial tissue that we're going to look at here in the lab, is this transitional epithelium. So here, again, they're using the urinary bladder, and I talked about this. This urinary bladder is going to fill with urine. It's going to expand like a balloon, filling with air, and that's going to create the need for extra additional surface area on this inner layer, inner wall of that structure. How are we going to get that? We're going to get it by stretching these cells out to become flat. So these cells are going to be going from cuboidal to flat, back and forth, depending on the condition of the urinary bladder. And they're designed to go through that change from flat to cuboidal, and back and forth without any damage. That's how they're designed. And as I said, you really have to develop an eye for this. And I will give you lots of images, and we'll go walk through them one at a time so you can see what we're talking about when we talk about transitional epithelium. Because you might think of this as stratified cuboidal almost. And yeah, but there's some differences. So first of all, the cell shape, the cuboidal shape, is structurally different. See, at the top, they're very large. Down here, they're smaller. You're not going to see that with stratified cuboidal epithelium. So the cell shape is going to be relatively the same throughout the column. You're not going to see different sizes of cuboidal shaped cells as you do here with transitional. Okay? And there's some other secrets that I can show you when we get into the images. Here is my basement membrane. You can see it quite clearly. That dark line is my basement membrane. Okay, and so everything up to this point is my epithelial tissue. Okay, underneath it, connective tissue. That's this layer here. Okay, so very thin basement membrane and then connective tissue. So very thin basement membrane, and then the rest is connective tissue. Okay. All right, ladies and gentlemen, that does it. I just want to very quickly turn back to the lab and kind of guide you to what we've done so far. So we've talked about epithelial tissue. We've introduced the structure, basic structure of epithelial tissue. We looked at different shaped cells and different layers. We've looked at this system of naming. Come back. There it is. Of naming epithelial tissue. And this is the list of epithelial tissue that you're going to be responsible for in the lab. I'm going to expand on this list a little bit more in lecture. But in the lab, this is what we're focusing on, because these types of tissues, simple squamous, simple cuboidal, simple columnar, and stratified squamous, and transitional to some extent, this is what you see most of in the body. This is where you're going to run into this type of tissue most often when you're going through the various organ systems. Okay, so I'm not showing you all of it in the lab, just the ones that are most common. And then I've broken it down. So here's my simple squamous. And here are some questions that you should be thinking about. Name 3 functions of this type of tissue. Name 3 areas in the body. And where would you not find this type of tissue? How are you going to find those answers? Well, you're going to look in your textbook. And in your textbook, there is, and I've noted the pages at the beginning, there is a table that lists each of these different types of tissues and gives you that information. I'm going to leave that up to you. So here it is. This is simple cuboidal. Again, name 2 functions, name 3 areas. Okay. So simple columnar epithelium, name 2 functions, name 3 areas. What is the advantage of this type of tissue over simple cuboidal epithelium for secretion? I want you to think about that. So that question is probably not in the textbook at all. But I want you to think about this. When we do our lab review, we're going to look at that question and address it. But now I want you to suffer a little bit. All right, so here it is, stratified squamous epithelium. Again, name, function, is type of tissue, areas in the body. And we're going to talk about keratinized versus non-keratinized. Again, I didn't talk about that. So you're going to have to do a little research on that, see if you can come up with an answer what that is. And when we do the review for this lab, we'll talk about it then. Okay? And then finally, transitional epithelium. So here are two images. So kind of difficult to see, but I'm going to come back to these images. I'll have these images in a larger format when we look at this. But here, if you see, this is very cuboidal shape, and now they're flattened out here. So flattened shell as opposed to a cube-shaped cell. Okay? So I'm going to transition from this shape to that shape, back and forth, depending on whether the bladder is filled or it's empty. Okay. And here, where is this type of tissue found in the human body? What is the function of this type of tissue? Okie dokies. So that's where you're going. You've got some work ahead of you. The next lecture I'm going to do, we'll post, will be images of epithelial tissue, and we'll look at those. I'll talk about them as we're looking at them, and we'll have a lot of fun. So see you next time.